Good morning, everyone. First of all, I would like to thank the organizers for inviting me here, and as usual, I'm very pleased to be in Greece. Today, I will try to talk on the evolution of clinical trials, and I will talk about three pathways of evolution. I hope I would. Okay, I will change this. Yeah, and I will talk on the regulatory evolutions, the methodological evolutions, and also the technological evolution. In 1974, a Scottish captain, Navy captain, took 8 ships, and he went to an overseas journey to the new continents. And at the beginning, there were around 1,800 sailors with him. But when he returned back to England 4 years later, there were only 188 sailors left. And most of these sailors died because of an flesh-eating disease. And at that time, there was a bright doctor, Navy doctor, James Lind, and he thought that if he could provide an acidic substance to the sailors, he can stop the disease. So what he did was, he took 12 sailors on the journey, he made 6 groups of 2, and he gave 6 different acidic substances to each group. And at the end, he found out that the group taking oranges and lemons, they did well at the end of 1 week. So he thought that he could cure the disease. Actually, this was vitamin C deficiency. And he published it as a paper, and every year we celebrate this paper as the International Clinical Trials Day. Well, this is a nice story to hear, but when we look at the history of the clinical trials, we always see that these regulations, these designs, methodologies, we had never been proactive. What happens is, a disaster occurs, and after this disaster, we had the regulations. After these problems, we had the new methodologies. But eventually, now we have 2 different important documents or regulations. The first one is the Declaration of Helsinki, which shows us the ethics of the clinical trials, and the second one is the ICH-GCP, which shows us how to perform a clinical trial. And most of the regulations and other things that we have now are based on these 2 documents. And after that, we began using the FDI-defined terminology, like the phases in the clinical trials. So we have a preclinical phase, then we start doing phase 1, phase 2, and we go on like that. And we notice, but in the last 10 years, we can see a tremendous change in this terminology, in the methodology, in the design of the clinical trials. What's the reason for that? Actually, now we are having a lot of emerging disease. The disease which spread out very fast, which kills a lot of people, so we need new drugs, but we don't have time to develop these drugs. And the second issue is, we have a lot of orphan diseases. And the problem with orphan diseases is, we have very few patients. So it's not easy to do a full-blown clinical trial in such diseases. And a new concept, personalized medicine or targeted medicine. This is now a new area that we have to look after. But eventually, at the end, the main problem is time and cost. We are spending 10 to 15 years to develop the drug, and we are spending a lot of money. This is from Tufts University, and taking everything into account, they found out that to develop the drug takes around $2 billion. Maybe this is an overestimation, but you can easily see that we spend a lot of money for that. So why not we shorten the time? Why not we decrease the costs of the clinical trial? And also another factor is, well, now the society is pushing us to develop the new drugs, to put them as early as possible on the market. I don't know if all of you watched this movie. This is the Dallas Buyers Club. It's about the AZT trial in AIDS. And try to watch this, and you can easily understand what I'm meaning by that. And also, in terms of the emerging disease, we also feel this pressure to have the drug as early as possible. But how can we do that? Well, we can do it in 2 ways. We can modify our regulations, and especially we can do it in the authority review process. And also we can modify our methodologies. And when you look at this part, when you go to the FDA review process, you can easily see that there are quite intelligent way to do that. So the FDA has a expedited review process, and there are 4 different pathways, like accelerated approval, priority review, fast track, breakthrough therapies, and they are very well defined. So if you are a sponsor, if you're a company who has a new drug, you can go to the FDA and tell them that you want to use one of these routes. But don't forget that all these programs are aimed at serious and life-threatening disease. And there is a strong evidence that when you are using these pathways, you can easily shorten the duration of the clinical trial. And also you can modify your methodologies. And there are several ways to do that. And I would like to talk on them. First, now we began using surrogate endpoints instead of clinical endpoints. Actually, a surrogate endpoint is a marker that can predict the clinical outcome of a disease as early as possible. So instead of using the golden standard, the clinical endpoint, you can use a surrogate endpoint instead of that. And that makes the duration of the clinical trial as short as possible. And actually this study started in the year 2000s after the discovery of a new drug called Gleevec, imatinib. Imatinib is a drug that cures a leukemia, chronic myelogenous leukemia. This leukemia is a chronic disease, and in the first 5 or 6 years, the patient has very few symptoms. But after 5 years, the disease changes. It changes to an acute leukemia, and the patient dies in 1 or 2 years at that time if he didn't have any transplant. So if you have a new drug that can cure this disease, to see the result of the drug, you have to wait for 7 years till the end of the trial. And it's a long time for a new drug to develop. But the lucky thing here is this disease have a molecular target, molecular marker, a surrogate endpoint. This is the Philadelphia chromosome. This chromosome produces a protein called BCR-ABL, and it's clearly shown that if you can suppress this BCR-ABL, you can cure the disease. You don't have to wait till the end of the 7 years or 10 years or something like that. But there are 2 rules to use something as a surrogate marker. First of all, the surrogate endpoint, the marker that we are using, has to be correlated with the clinical outcome, has to predict the clinical outcome. And the treatment we are using should have a correlation between, should have an effect on the surrogate endpoint and the clinical outcome, and this has to be correlated. And if you look at this article that was published in JAMA in 2017, you can easily see that if the drugs, 2/3 of the drugs approved by the FDA are approved based on a surrogate endpoint, not on a clinical outcome. So this is the way things are going now. But don't think that you always have to have a molecular marker for that. We have also surrogate markers for classical disease. As an example, you can use hemoglobin A1C for diabetes mellitus. You can use blood pressure measurements for hypertension disease. So we have a lot of surrogate endpoints. And at the end, this promoted us to develop new biomarkers for diseases. So people are now working on developing new biomarkers. And this is taken from the FDA webpage. You can see that we have a lot of surrogate endpoints. Now we are using them as the outcome. And this is a good example. This is a trial on CML, the disease I mentioned. And instead of waiting for 7 or 10 years, this study took only 24 months, 2 years to complete the trial. So we can give a lot of experience like that. Another different methodological approach is the basket trials. These basket trials are quite ingenious. There are 2 ways to do that. You may have a single diagnosis, lung cancer, with a lot of mutations, or you may have different diagnoses, different cancers, with the same mutation or with multiple mutations. So in one trial, you use all your drugs, and you include all these patients in the trial. This is a good example is the IMPACT study. In this study, we have 3 different diagnoses of patients, and we put all of them in a trial. And now we have a lot of patients in the trial. And there are different mutations, and we use different drugs against them. If one drug fails, you left the patients out the study, and you continue with the remaining patients. So these basket trials are a good way to go. Different tumors and different markers. And one of the most important developments are adaptive designs. This concept originates from this. What we usually is we complete the trial, we publish it, and we learn a lot about it. But we realize that we can even learn about something when doing the trial, not at the end of the trial. So this is called learning while trying or working. And what we do is we can modify one or more specified aspects of the study design or the hypothesis based off the interim analysis. But don't forget that. You have to do it before you start the trial. You have to decide everything before you start the trial. You have to write your protocol according to this, not in between the trial, to have an adaptive trial. So this goes on like that. First, before starting the trial, you decide on your time points of the analysis, interim analysis. You decide how you are going to do the analysis, and you decide what you are going to do after you receive the analysis. So you write the protocol, you do the interim analysis depending on something, and after you solve the results, you modify your trial. You modify the number of patients, you modify the randomization procedures, you modify your endpoints, whatever you want. But you have to write it at the beginning, not during the trial. And these 2 basket trials and adaptive trials have this advantage. Now you can get your drug approved after phase 1 or phase 2. You don't have to wait till the result of the pivotal studies, the phase 3 studies. So this is a good example. Ceritinib, it is a drug against lung cancer, and FDA approved it after phase 1 and after phase 2. These are called conditional approvals, of course. I mean, still they are looking for phase 3 results, but you can get your drug approved as early as possible. And during the Ebola disaster, these are put in place to have the vaccines as early as possible. And another important approach is, which is very popular nowadays, the real-life data, or what we call big data. The problem with the clinical trials is they don't reflect the real life, because we are using at most 1,000 or 2,000 or 3,000 patients, but when you go to the population, there are a lot of millions of people with these diseases. So what can you do? You can do mega trials. That means you can do clinical trials with 10,000 or 20,000 people, but the problem here is you need a lot of money for that. And it takes a lot of time, it takes a lot of organization, and it takes a lot of new techniques to do that. So, and at the end, you are never sure that this reflects real life. So what we are doing now is we are looking at patient databases, and we are trying to get some data, some meaningful data out of it. And we are using big national databases, we are using patient surveys, patient monitorization reports, the cohort studies, and disease registries. And we are trying to combine them with the results we take out of the clinical trials. But there are also problems with the big data, real-life data. You don't have these databases everywhere. I mean, if you are living in Holland, well, you have it. If you are living in Sweden, you have it. Switzerland, you have it. But in Turkey, you don't have a lot of big databases. And also, the quality of the data is a very big problem. The data can be ambiguous, the outcomes can be ambiguous, and there will be a lot of missing data. So you are never sure about the results you have, and you will always have some confounding factors and also bias in such data. And the last thing is, we also had a technological evolutions. In 1980s, what we have technologically, what we have at hand is a landline phone, a fax, and if you are lucky, you would have a photocopy machine or a scanner. But in 1990s, the computers entered the clinical trials. And we started to use the computers in doing our clinical trials, and at the beginning, we had this interactive response technologies that we can we can use the computer to randomize a patient and things like that. But in the beginning of the year 2000s, we had these systems more organized and more effective. But most of the systems that we use at these years are isolated systems, only for one clinical trial. But nowadays, we are having integrated systems like single sign-on, like e-sources and things like that. And another important advancement in this area is the Internet of Things technologies. Maybe you have heard about it, but last week, FDA approved this device. This is actually a wristband connected with your iPhone, and you can, by using your two fingers, you can take your electrocardiogram. And the ECG is recorded to your watch or to your iPhone, and you can send it to your doctor. So I think this type of technologies will improve and will take a lot of things in the clinical trial process. And of course, there are a lot of things that has to be talked on this subject, like enrichment studies, event-driven trials, and composite endpoints. But for time's sake, I'm going to stop my talk here, and thank you very much for listening to me.